The greater white-fronted goose is a species of goose. The greater white-fronted goose is closely related to the smaller lesser white-fronted goose. In Europe it has been known as simply white-fronted goose. In North America it is known as the greater white-fronted goose, and this name is also increasingly adopted internationally. It is named for the patch of white feathers bordering the base of its bill, but even more distinctive are the salt and pepper markings on the breast of adult birds, which is why the goose is colloquially called the speckled belly in North America. Answer is the Latin for goose, and albifrons comes from the Latin albus, white, and frons, a forehead. Description: greater white-fronted geese are 64 to 81 centimeters in length, have a 130 to 165 centimeters wingspan and weigh 1.93 to 3.31 kilograms. They have bright orange legs and mouse-colored upper wing covers. They are smaller than greylag geese, as well as being larger than the lesser white-fronted goose. The greater white-fronted goose lacks the yellow eye-ring of that species, and the white facial blaze does not extend upwards so far as in lesser. The male is typically larger in size. Both sexes are similar in appearance, grayish-brown birds with light gray breasts dappled with dark brown to black blotches and bars. Both males and females also have a pinkish bill and orange legs and feet. Differences between European and Greenland birds. The appearance of European or Russian white-fronted geese of the race albifrons and Greenland white-fronted geese of the race Flavorostris differ in a number of ways. The Greenland white-fronted goose, in all plumages, looks darker and more oily-looking than the European white-fronted goose, both at rest and in flight. The following are the differences which apply to first winter plumage. The mantle and scapulars of Flavorostris have narrow, indistinct pale fringes creating a uniform appearance to the bird's upper parts, whereas albifrons has noticeable whitish fringes creating obviously barred upper parts. The tertials of Flavorostris have indistinct pale fringes, whereas these pale fringes are more noticeable on albifrons. The lesser and median upper wing covers of Flavorostris have narrow, indistinct pale fringes, creating a rather uniform appearance to the wing, whereas on albifrons, these fringes are prominent and broad, creating wing bars. The greater covers of Flavorostris are dark gray, with a narrow white tip, forming a narrow wing bar. On albifrons they are blue-gray, with prominent white tips, forming a bold wing bar. The flank line is narrow and white on Flavorostris but broad and bright white on albifrons. The tail of Flavorostris is dark brown, with a very narrow white tip and sides. That of albifrons is dark gray, and the white tip and sides are at least double the width of the corresponding areas on Flavorostris. The bill of Flavorostris is orange-yellow with a dark nail, compared with the bright pink bill of albifrons which has only a hint of dark on the nail. In addition, the bill of Flavorostris is longer and appears slimmer than that of albifrons. The belly barring on adult birds is on average more extensive on Flavorostris than on albifrons, but the individual variation in both forms renders this of limited use as an identification feature. The bill of adult Greenland white-fronts are also orange-yellow at the base, but can be more pinkish-yellow on the outer half, thus close in color to European white-fronts. The color difference is more easily determined in dull, flat light rather than bright sunshine. Taxonomy. The greater white-fronted goose is divided into 5 subspecies. The nominate subspecies A albifrons breeds in the far north of Europe and Asia, and winters further south and west in Europe. 3 other restricted range races occur in northern North America. A gambelli in interior northwest Canada, and wintering on the coast of the Gulf of Mexico, slightly larger than the nominate form. Pacific white-fronted goose (A. frontalis) and Tule goose (A. elgazi) in southwest Alaska, largest and longest-billed of all, both wintering in California. All these races are similar in plumage, differing only in size. The very distinct Greenland white-fronted goose (A. flavirostris) breeding in western Greenland is much darker overall, with only a very narrow white tip to the tail, more black barring on its beak, and usually has an orange bill. It winters in Ireland and western Scotland. Birds breeding in the far east of Siberia east to Arctic Canada, wintering in the United States and Japan, have been described as A. frontalis on the basis of the slightly larger size and a marginally longer bill. Another putative East Asian subspecies, albicans, has also been described. A 2012 study has found that frontalis and albicans do not merit subspecies status, the former being synonymized with gambelli and the latter with the nominate subspecies. This study found that these forms had been named on the wintering grounds from specimens whose breeding grounds were unknown. Ecological studies in 2002 suggest the Greenland birds should probably be considered a separate species from A. albifrons. Of particular interest is its unusually long period of parental care and association, which may last several years and can include grandparenting, possibly uniquely among the Anseriformes. Distribution. The North American mid-continent birds of the subspecies A. gambelli, which in 2010 had a full population of about 710,000 birds, breeds from the Alaska North Slope across the western and central Canadian Arctic. The Pacific white-fronted goose of the American Pacific coast, which in 2010 numbered approximately 650,000 birds, and the Tule geese, which are estimated to number 10,000 birds, nest in western Alaska. The mid-continent geese gather in early fall on the prairies of western Saskatchewan and eastern Alberta, spending several weeks feeding before heading to wintering areas near the Gulf of Mexico, into northern Mexico. The Pacific birds migrate south down the Pacific coast, staging primarily in the Klamath Basin of southern Oregon and northern California and wintering eventually in California's Central Valley. The Tule goose is somewhat rare and has been since the latter half of the 19th century. Presumably it was affected by destruction of its wintering habitat due to human settlement. In the British Isles, two races overwinter: Greenland birds in Scotland and Ireland, and Russian birds in England and Wales. They gather on farmland at favoured traditional sites, with a famous flock gathering at WWT Slimbridge, Gloucestershire, England. Greenland birds also overwinter in Ireland and from late September and through the winter months. Ireland is home to almost 50% of the Greenland population of white-fronted geese. A. albifrons and A. flavirostris are among the taxa to which the Agreement on the Conservation of African Eurasian Migratory Waterbirds applies. Behavior and ecology. Weather conditions are a key factor in the annual breeding success of white-fronted geese. In the Arctic, the window of opportunity for nesting, incubating eggs, and raising a brood to flight stage is open briefly, for about 3 months. Arriving in late May or early June, white-fronted geese begin departing for full staging areas in early September. This means that a delayed snow melt or late spring storm can significantly reduce the bird's reproductive success. Origin of migration. Mid-continent white-fronted geese have many breeding area from North America, and each white-fronted geese group in each breeding area has differences in migration time and the location to winter. There are 6 breeding areas, including 1 interior Alaska, 2 North Slope of Alaska, 3 Western Northwest Territories, 4 Western Nunavut, 5 Central Nunavut, and 6 east and Nunavut. These spatial differences lead to time differences for white-fronted geese to leave the breeding area and differences in location to winter. Birds from interior Alaska start migrating earlier during autumn and fly farther south to winter, fattening before migratory flight. Breeding environment has a large impact on the success of migratory flight, which include the temperature of the environment. Migratory bird that has faster fattening rate when foraging in colder environment. Backtracking technique. Ways to track migratory path of greater white-fronted goose have been the same for most research. There is one research that does not only look at the collective data from the whole population but an individual. This researcher from Austrian Institute of Technology named Misha Horacek believes types of feather growing on a single migratory bird can be a way to backtrack the migratory route of a migratory bird that was infected by bird flu. Therefore, once a migratory bird is found sick, the affected area can be measured and marked. Bird feather grown in a timeline which can be used as timeline for nutritional intake during the migratory flight. This allows people to understand the route which the infected bird took and determine the region affected by the particular bird flu. During migratory flight, feathers on white-fronted geese gradually fall off during migration as new feathers grows. This renewing of feather become a record book for the migratory journey. Every environment has its own distinct signature that is recorded in isotope signal of carbon, hydrogen, nitrogen and sulfur isotope level, and these signal were picked up by migratory birds by ingesting native resources. Even though food were digested, the basic element still has isotope signal that tells people where these elements are from. Feathers were collected, washed and dried with an oven at temperature less than 45 degrees Celsius. Then various measuring instruments were used such as thermal combustion elemental analyzer for hydrogen and Vario 3 elemental analyzer for carbon, nitrogen and sulfur. These signals are recorded onto the feather that were growing during the migration. There are different types of feathers on great white-fronted geese that have different growing rate, and these growing rate can be seen as timetable. As to the time each isotope signals were picked up, this isotope signal can be analyzed and compared to the isotope signal collected from each environment. This allows researcher to understand the migratory path of an infected bird and estimate the affected region within the migratory path. The result include extreme depletion in 2H on primary feathers, which represent the environment condition in Arctic Siberia. However, more analysis needs to be done on feathers of local bird species in various environment, which allows more comparable data to match with feathers of migratory bird. Gallery.